Hi, I'm Mauricio from Minitube Brazil. Today in this video I will talk about some important details in the sample preparation for semen analysis on Androvision CASA system. So Androvision is a highly precise and accurate system for the computer-assisted sperm analysis. This is where CA-assay initials came from. Androvision includes all the accessories needed to carry out the sample preparation. The sample is then placed in the microscope, which has a camera connected, and the live image is analyzed by the computer with software, which is the Androvision itself. So the main focus of this presentation will be on the sample preparation for standardized motility and concentration measurements. Before talking about the procedures, it's important to point out that the system accuracy and precision depends on how the technicians operate the system. So the technician responsibility is determinant to ensure the correct use of the system. Well-trained technicians will provide reliable information for trustable data analysis afterwards. Considering that a correct operation of the system can lead a very low coefficients of variation, some procedure mistakes may cause an increase in the variation of results. That's why sample preparation is so important. In fact, the procedure is quite simple. We could sum it up in just 6 steps. First, take the extender. Second, take the semen. Third, make an air bubble. Then wipe the tip. Dispense the sample in a pre-warmed vial. And then finally, load the counting chamber. And why we need to do this pre-dilution procedure? To have an image like this, with individualized sperm cells in the field to be analyzed by the CASA system. So although the process is very simple, we need to understand what we are dealing with to avoid make some mistakes that could negatively impact the results of the system. Extender at low temperatures. It's known that temperature is very important when handling semen of any species. Normally the temperature should be at around 37 to 38 Celsius degrees. In this example, if we could look with the super thermal vision glasses, we would notice that the desired temperature actually is only at the bottom of the vessel. At the top, the extender is cooler, and this is where you will take the sample from. When you put this cold extender in contact with the semen sample, you can cause a thermal shock, and many sperm cells will be killed. This is an example of what happens when the semen suffers a cold shock. Typically, many dead sperms, most of that appears with bent tails, and the live ones have this lateral head movement and circummotility. So always choose to work with small containers rather than bigger ones. And also replace the extender from time to time if you have it for long periods over the heating plates. Loose tip in the pipettes. This is a small detail that few people are aware of it. Normally, take the pipette tips from the boxes is so automatic and fast that we cannot imagine that this can represent a source of variation in the results. And then I will show you why this can happen. Look to the end of the pipette and a dashed line right in the middle to be our reference. Then here is our pipette tip, and this represents when they are perfectly attached. When you aspirate the liquid, nothing happens. So now let's change a little bit the position of both parts to represent when you don't tighten them enough to fit them perfectly between each other. So then, after you aspirate the liquid, you have no control over these leakages. And of course, lose 1 or 2 drops of the sample can represent a big issue for the precision of your results. Tip immersion depth. Immersing the tip to the correct depth will improve your accuracy. When positioning the tip too close to the surface can aspirate air. Conversely, immersing the tip too deeply can cause too much liquid to be aspirated. Depending on how much you immerse the pipette, you can even have this situation when the semen or foam reach the pipette itself. In this case, your pipette will lose accuracy over time. Failure in mixing the ejaculate. One of the first steps in the sample preparation is the mixing of the ejaculate, and many people fail in this primordial step. Here an example of an ejaculate, only to show you what happens if you keep it static for longer than a minute. You will have a sedimentation of sperm cells at the bottom of the container where the semen is. And this will result in a different portions in your ejaculate, where the upper part has a lower sperm concentration than the entire ejaculate. And this is where you will take the sample from. So to avoid this basic but common mistake, you only need to remember to always mix the ejaculate by rotating it at least 5 times upside down, but please, no shaking. Bubble suction. Shaking the ejaculates too much would lead you to another mistake, which is the bubble suction. In this picture, you can't even see where the semen surface is. So gently mix the ejaculate to facilitate obtaining the sample. Semen residue outside the tip. When immersing the tip in the ejaculate to get the sample, you will automatically have semen residues outside the tip. As we are always working in a vertical position, this semen residues tends to accumulate at the tip end. That's why we recommend to wipe the tip as per talked before. If that goes along with the sample, it can cause a significant variation in the results. So let's make a calculation exercise with this drop of 5 microliters. Let's consider that it happened in a bull semen, using a standard dilution ratio of 1 plus 29, 30 million sperm per dose, and mini straws. Well, 1 plus 29 dilution ratio means 25 microliters of semen plus 725 microliters of extender. In this example, if we have an ejaculate concentration of 1.2 billion sperm per ml and an ejaculate volume of 10 ml, then we would have 12 billion sperm in this ejaculate. This would be enough to produce up to 400 doses, and we would need to add 90 ml of extender. So this is the correct procedure, and we would have our desired 30 million sperm per dose. However, if we had included this unwanted drop in our sample, it represents 20% more semen than the correct amount, and we would have 30 microliters of semen. So CASA system don't know that and would give us a result of 1.44 billion sperm per ml. In the same ejaculate with 10 ml, it would result in 14.4 billion total sperm in this ejaculate. And after the dose calculation, this would produce around 480 doses, and we would need to add 110 ml of extender. It's more extended than really needed. Look that we are wrongly processing 480 doses, whereas we have, in fact, only 12 billion total sperm in this ejaculate. This effort would end up causing a reduction in the sperm concentration per dose. So yeah, this drop represents, in this example, 2.4 billion more total sperm in this ejaculate, 18 more produced doses, and 5 million less sperm per dose. Hence the importance of the sample preparation. Sample homogenization. Take care to assure a constant workflow in your lab. If the sample was kept vertical for longer than 1 minute, it has to be rotated again upside down at least 5 times before take the sample. And again, don't shake as we saw earlier. In the other hand, it doesn't mean that you need to do it so slowly like this. Otherwise, you can see this effect when the liquid in its first contact with a dry tube gets stuck on the plastic wall and actually doesn't mix anything. Pipetting cycle. The pipetting cycle consists of 5-step process. First, push down on the plunger button until you feel it reach the first stop, and hold. Now, immerse the pipette tip in the sample, and then aspirate liquid into the tip by slowly releasing the plunger, allowing the plunger to return to its home position. Then dispense the sample out of the tip by pressing down on the plunger in a smooth, continuous movement until it reaches the first stop. And now we do the blowout by pressing the plunger until a second stop. It spells any sample remaining in the tip. This drop that forms at the end of the tip is the one that will load the counting chamber. Only after fill the chamber you should release the plunger to avoid aspirate the sample again back to the tip, and then finally eject the tip. Pipetting speed. To ensure that you are achieving the highest accuracy, keep constant speed and smoothness when pressing and releasing the plunger, and avoid too fast movements on the pipette plunger that can cause liquid splash and aspiration of air. Pipetting angle. Pipetting with the incorrect immersion angle is one of the most common incorrect pipetting techniques. In fact, we recommend to keep the immersion angle as close to the vertical position as possible. Drop size. It depends on which counting chamber you use. If you use mini-tube counting chamber, then you should use 2.5 microliters at the pipette. If you use Leia counting chamber, then you should use 2.7 microliters at the pipette. This can change a little bit according to the calibration of your pipette. The most important in this case is if you are able to fill the counting chamber with the exact needed volume in only one single step. Here we have two situations, an excessive volume on the left and a corrective volume on the right. If the drop size is too big, look what happens. The sperm drift is caused when redundant liquid stays in the filling area of the chamber. This liquid keeps on pressuring into the chamber by capillary forces and causes the drift of the sperm in the chamber. In this case, you would see these very long lines across the field and maybe higher motility results. On the other hand, if the drop size corresponds to the exact volume needed for the counting chamber, then all dead sperm cells are really static in the field. So the motility results would represent the reality in this case. Incomplete loading of counting chamber. This is other very common situation that happens. It can be caused if a drop size is too small or even if part of the sample remains into the tip. If it happens to you, please don't try to improvise by adding a little more liquid to complete the chamber. Instead of this, fill a second chamber to get the correct results. Active loading of the counting chamber. The counting chamber should be filled in a passive way. So press the sample volume out of the tip before the tip reaches the chamber. As shown before, a small drop appears at the end of the tip. And then place this drop in the filling area of the chamber and it will fill itself by capillary forces like in this video. This procedure will ensure an uniform sperm distribution through the chamber, which is good for sperm concentration measurements. Time between sample pipetting and chamber loading. This is a very critical point mainly for motility. It's not recommended to take the sample directly from the heating block because you cannot really see where the sample is. It's also not recommended to keep the counting chamber always in the reading position at the microscope stage because it is an area that the counting chamber does not have direct contact with the warming plate. So people do it to wrongly save time and labor. But in the end, this long time span between take the sample and reach the counting chamber is sufficient to considerably decrease the motility of the sample. Thus, look at the needed pipette angle in this case. It's wrong as well. The recommended procedure consists in put the counting chamber previously on the heated part of the stage. Position yourself closer to the microscope, then take the sample and immediately load the chamber. This will improve your motility results a lot. Reading area. Only use the central part of each chamber to take the fields that you need in your analysis. Avoid measure areas close to the edges because sperms tend to accumulate in these areas, disturbing your motility and concentration results. Besides that, start a first field from the upper part of the chamber, which is far from the filling area, and then go down analyzing the other fields. This will prevent you from analyzing the same sperm cells. External dirt on counting chamber. Labs that don't properly clean the heating plates and the counting chamber prior to analysis will see these glowing circles on the screen. Sperm cells that are right over these circles during the analysis may not be detected by the system because it's different light intensity in comparison with the rest of the field. If you keep all this stuff properly clean, you will not have this issue. Consumables reuse. We do not recommend any reuse of pipette tips and other disposable materials. In this example, it's possible to notice that there are water residues inside the tips because it was washed and not properly dried. So of course the motility results will be worse in this case since water is spermicidal itself. So look how important is the sample preparation for the accuracy and precision of your CASA system, and also the technician responsibility in all this process to generate reliable information for further data analysis. Well, hope this walkthrough, sample preparation and pipetting techniques can help you to improve your results with Android Vision. If you have any questions, please don't hesitate in contact us. One of our specialists will get in contact with you. Okay? Thank you. Bye bye.